Can I do that later? Okay, I'm not saying do it.
All right, guys. I'm gonna go ahead and take attendance, and then we'll get started. We have a lot to cover today. Briley? Here. Brianna Akins? Here. Andrew?
Here.
Bria?
Here.
Henry?
Here.
Anna? Anna B? Nick?
Here.
Abby?
Here.
Hannah?
Here.
Rosa?
Here.
Matt?
Here.
Jillian? Mia? Annie? Annie? Brianna Hills?
Here.
Brandon? Sadie?
Here.
Jessica?
Here.
Jesse? William? Marissa?
Here.
Anna? Sophia? Aubrey?
Here.
Matthias?
Here.
Olivia?
Here.
Braden? Amber?
Here.
Katie?
Here.
Kennedy?
Here.
Ava?
Here.
Kendall?
Here.
Emily?
Here.
Lauren?
Here.
Jen?
Here.
And Isabel?
Here.
Okay, awesome. So we stopped on Wednesday. We stopped over here, so I'm going to go through just some more microbial diversity, talk a little bit about fungi, and then we'll dive into chapter 2, because the stuff we'll talk about in chapter 2 are going to be very relevant to what we're going to be doing in lab next week. So fungi are microbes as well. They're eukaryotic. There's so many different species, and they don't carry out photosynthesis. They're fungi. One key feature about fungi is that they have cell walls similar to plants, but the thing that the cell walls are composed of is called chitin, and it's a really, really strong polysaccharide. So chitin in cell walls is something that we'll talk about more, and we'll look at some of their structures under the microscopes in lab. So their membranes contain ergosterol. You guys don't need to know that for this class. But one of the important things about fungi is that they can come in different forms. They can be unicellular or they could be multicellular forms. So typically the single cells that we see here, we call those yeast cells. So that's usually used interchangeably with fungi. But fungi can also, form molds. The bread mold that you'll see on your bread is actually a fungus. So that's a multicellular, or what we call a mycelial mold. Some fungi don't just exist in one form or the other. Some of them are able to exist in multiple forms. And the one that is really well known, that I actually studied for my dissertation, is Candida albicans. Anybody heard this in the context of health, the yeast Candida albicans? So yeast infections are due to this fungus, Candida albicans. And its ability to have these two forms is implicated in its ability to form yeast infections. It's a normal member of the microbiome. It exists in us. And so it doesn't normally cause harm, but it is the causative agent of these opportunistic infections. So it can become pathogenic. So it's usually the cause of yeast infections and oral thrush. So it's dimorphic, so it can form as these single-celled yeasts, but then it can also form these filamentous hyphae that we also call mycelia. These are actually images that I took, and a little experiment that I did at one point was to see, the hyphal formation over time in Candida albicans. So this is normal growth media for yeast, and so you can see that you have these yeast cells, and then over time you start to form these hyphae, but then there is certain triggers that can trigger hyphal formation, and so serum is one of those things that we know triggers things to grow and form hyphae, and so when I put the fungus in the same media but with this 10% serum, you can see that just by 30 minutes it started to change to the filamentous form. So why do we care about this? Well, the ability of this fungus to switch between these forms is actually a defense mechanism that it uses to invade or escape the immune response. But in fungi, generally, there's so many different species, and so they show very different forms. So they can look like the mycelia or the filaments that I just showed you. We also call those hyphae. They could look like fruiting bodies, mushrooms. They also could be in association with plants, and so we call this one mycorrhizae. And so myco means fungi or fungus. That's why the study of fungi is mycology. And rhiza means roots. So mycorrhiza is the symbiotic association between fungi and plant roots. So if you see here, there's this fungus, and so it has these filamentous hyphae that are in the ground, and here's a tree, and so you can see that they make these associations with the tree roots, and there's this is a symbiotic association, so there's obviously implications to that, and it's usually beneficial to have this sort of association. So what's the benefit of this plant mycorrhizal relationship? Here's a plant without the mycorrhizal fungi, and here's another plant with the mycorrhizal fungi. So you see that the plant has grown a lot more. So what are the benefits that the fungus is providing to the plant? Well, because the fungus is able to connect with the plant roots, it's able to extend the volume of soil that's accessible to the plant through the hyphae. So it provides increased minerals for the plants, and it also increases the plant's access to water. Because plants need water, they need nutrients, so this helps also extend the volume of soil that's accessible to the plant through hyphal transport of water. So here you can see what it looks like underground. Not only do you have this plant root system, but you also have this hyphal system that also allows, gives the plant an extension to reach more nutrients and water. Questions so far? Okay, well, in a symbiotic association, it's a two-way street. So, for them to be able to be associated like this, the plant and the fungus, well, there's got to be benefits to the fungus as well. So what does the plant provide to the fungus? Well, it provides a dependable and abundant source of carbohydrates because, as you know, plants undergo photosynthesis, and you all should have learned that one of the results of photosynthesis is you make glucose or carbohydrates. Fungi don't photosynthesize, so this is a really amazing source for carbohydrates, and they get it from the plant. So this is this classic mutualistic symbiotic relationship where both parties are benefiting. That's not always the case, but in this case they are. And so now I'm going to just introduce one of these really, really cool organisms that's called a lichen. A lichen is not just one thing, it's a composite organism that arises from an association between two things. Usually it's an algae and a fungus, or it's a cyanobacteria and a fungus. So we talked a little bit about algae and cyanobacteria. Remember, cyanobacteria are sometimes called blue-green algae, but they are not algae, they are bacteria, but there are similarities. So this lichen is a very specific pairing of a fungus and an algae, or a fungus and a cyanobacteria. And so they also exist in this mutualistic symbiotic relationship where both of the parties are benefiting. But the lichen, as the composite organism, is very different from the algae or the fungus growing alone. So it's got to have this association for it to be called a lichen. Lichens secrete organic acids. They weather rock. They're usually found on rocks like this, but they're really, really slow growing. So what does this look like if we were to slice a lichen? If you take a cross section of it, you'll see that there's this outer layer of this is the fungus, and so this is where the fungus the cortex, the outer layer, but then you can see that these filaments or these hyphae are really nicely and tightly wound in this area, the medulla. That's where the hyphae are kind of packed. But within those hyphae, you'll see the photosynthetic part of this association. And so here, this is the green algae that are photosynthetic. We know cyanobacteria are also photosynthetic. So you usually have, this loosely packed hyphae and then the tightly woven layers in the cortex and the lower cortex. And then these are also part of the fungus that just anchors it to the rock or whatever that substrate is. So just to give you guys an idea of what it looks like, you don't have to memorize these words, but just know what a lichen is. And so also understand what each partner receives from this relationship. So the algae, remember, is photosynthetic. So usually the algae gets increased water availability from the fungus because the fungus has the hyphal root system. Also helps from drying out because the fungi, provide access to water. And the algae then has a surface to attach for growth because normally it's not going to be able to attach to the rock or whatever substrate it is without the fungus. And what does the fungus get? It gets nutrients derived from photosynthesis. So usually the fungus will be in an otherwise nutrient-poor environment, so the algae provides that to the fungus. Does that make sense? All right. So just to kind of recap this whole microbial diversity lecture, what roles do microbes play in our everyday existence? And so these bullet points are according to very active and emerging research fields. So microbes, they play defense. They protect us from pathogens simply by just taking up space. And so they try to make sure that they take up the space so that it's not, open for invaders. Microbes also boost the immune system. Microbiology is very closely linked to immunology. And so bacteria can bind to immune cells and stimulate them, and then the immune cells can divide and reproduce and then fight that bacterial infection if something is going on. Microbes also protect us from autoimmune diseases because they help to train your immune system. Autoimmune diseases are diseases where your body cannot recognize self from non-self. So your body starts to attack itself. Your immune system thinks that something in your body that's normal is foreign, and so it will try to attack itself. So microbes help to train your immune system against that. They also influence your physical health. Microbes tend to, having a healthy microbiome is associated with decreased, obesity and things like that. So they help us to digest food, ferment them. They help shape our metabolism. They're really important. So when people say you are what you eat, that's very true, and it has a lot to do with the microbes that are feeding off of what you're eating. Microbes detoxify and may even fight off stress. And so this is an active research field, and so they help to take in toxins and they spare us the dangerous effects of toxins. And so microbes keep babies healthy. Babies that are born via C-section actually have very different microbiomes from children who are born vaginally. And babies born via C-section are more likely to develop asthma and allergies over time in life than children that were born vaginally. And that's because the microbiome that's transferred from the mom to the baby through the vaginal canal actually helps to prime the immune system of the baby, and it makes a big difference in the long run. The same thing goes for breastfed children versus formula-fed children. Breast milk is one of the main sources of that really healthy microbiome that children get from the mom. So this is, very interesting area of research. In my advanced microbiology class, that's what we're going to be talking about, reading papers on this. So overall, though, microbes are so important, and remember, we have an equal number of microbial cells as human cells in and on us, so they're constantly playing roles in our everyday existence. But it's not just about us. They kind of rule the world. We're just visitors. They're essential for life on the planet. They're involved in carbon cycling, nitrogen cycling, production of oxygen, and just, cleaning our environment. So you guys can read this on your own, just some more, things that microbes are involved in that have nothing to do with us directly, but they affect the world around us. All right, so I'm going to switch over now to chapter 2. All right, so chapter 2 talks about microscopy and how we observe the microbial cell. And so in lab, we're going to be using microscopes a lot. So I wanted to make sure that we understand how microscopes really work, because they're really important for our understanding. So we'll start by understanding a little bit about the physics of light. And so we'll talk about some things like that. And we'll try to get through staining today, because we're going to be staining bacterial cells in lab next week. So microscopy has relieved that we are just as microbial as we are human. So if we don't limit ourselves to just being human, you'll find microbes everywhere. And so we live in a microbial world, and bacteria are really beautiful. So the only tools that we have to see microbes are our eyes, so we need microscopes to help see microbes. And the size at which objects become visible depends on the resolution of our eyes. The resolution of our eyes is 0.1 millimeters. That means that that's about the size that we can see an image clearly using our eyes. So this is a very simple diagram. The smallest distance between these two red molecules is basically, in order for them to be separated, as independent circles is resolution. And so there's different things that limit our resolution that I'm not going to get into. But one of the things that we need is also detection. So resolution differs from detection. So detection is the ability to be able to determine the presence of an object. But resolution is being able to actually differentiate the different individual cells. So here we're looking at bacteria all over this plate. When the unaided eye detects this mold or bacterial colonies, that's detection, but you can't resolve the actual distinct cells. So that's kind of the difference there. There are tens of thousands of cells here that are making these bacteria detectable. But to be able to resolve or get resolution, you need to look under a microscope. And same thing with this plate that has colonies. Each one of these colonies is not an individual cell. It's tens of thousands of cells that allows you to see it as a colony. We call those a colony forming unit that's made from identical clones of tens of thousands of cells. So we also need more assistance to resolve the microbial cells, and that's magnification. So that increases the size of these small objects in order to be able to separate them. So just some terms. And so microbes range in size by orders of magnitude. So this is just an idea of the type of scale size that we're working with when it comes to microbes. You'll see that Prochlorococcus, Marinus, and Staphylococcus aureus, these are both bacteria, E. coli, and Bacillus megaterium. So Staphylococcus aureus, E. coli, and Bacillus megaterium are the three that we're going to work with in the lab, the majority of lab. So you can see that Staph aureus is smaller than E. coli, and E. coli is smaller than Bacillus megaterium, but they're still really, really small. So remember, this Prochlorococcus is 0.4 micrometers, and what was the resolution of our eyes? What were we able to see? 0.1, yeah, so it's just a little bit bigger, so that's why you still need a microscope even for the smallest microbes. So these are the three that we'll work with. Also notice differences in their shape. So these are cocci, that's why they have coccus in their name, which means that they are spherical shaped. And then these two are bacilli shaped, so that's where bacillus comes from for this name. And then this one also, notice that it's called Bacillus megaterium. The mega here refers to the fact that it's really large. It's known to be a larger bacteria. So a lot of things can be told from their name. Here's a little video just to help put this into perspective for you guys. These are viruses. They're really, really tiny. Still on viruses. Now we have some bacteria, a red blood cell, a yeast, now a skin cell, human sperm. Now these are things you can see, a neuron, human egg, yeah. All right, and all of that is sitting right next to a tiny hair. So really, really small. Okay, so microbial shape is really important. I mentioned some of these right now, but this is, certain shapes that are common to many of these bacterial groups. Cocci are spheres, bacilli have that rod-shaped, and then there's spirili, which are these rigid spirals. So sporuli are right here. They're a little more rigid. There's also spirochetes, so they're a little bit more flexible spiral bacteria. And then appendaged organisms have these like appendages coming out of them. So this is hyphae that would fall under appendages, and so they have these protrusions. So knowing the names of these shapes is really important. Any questions so far? Oh, cool, this is finally working. All of last semester and it didn't work. Okay, so just make sure. So microbes are generally larger than bacteria, or than eukaryotic microbes are generally larger than bacteria. So just another quick, differentiation there. Reminder of what prokaryotes are, what falls under eukaryotes, but they're all microbes. Of course, there's exceptions to every rule, though. But here are the different shapes. You have these rods. These are bacilli, and then you have a spiral right here that's next to a human blood cell, and then E. coli tends to kind of link in pairs, so these are cocci or diplococci if they're in pairs. So just some more shapes. Some more spirals, more chains of cocci, and more rods. So there's a variety of techniques in microscopy that allow us to probe the microbial world at different scales. So remember, the human eye can detect 0.1 micrometers. There should be a smaller m or a u essentially. And then we have light microscopy that can see up to 200 nanometers. And then we have other forms of microscopy like scanning electron, transmission electron, and others that can help you see even smaller and smaller microbes and help you see them different resolutions. So one of the things that I want to, and you don't have to memorize these numbers here, but one of the things that I do usually in these classes is I do little concept checks where I, just taught something and I just want to make sure that these concepts are making sense. So kind of just pausing here for a second. Which of the following statements about the size of microbes is false? Take a second to read this. You can talk with your neighbor, and in like a minute we can just discuss. Which is false. And this one's a little bit of a tricky question. Yeah.
Is bacteria plural?
It doesn't matter. Bacteria, I guess, in this case it would be plural, but it wouldn't matter. You can use it, like bacteria is used singular and plurally. Mm-hmm.
But like in this?
In this case, it would be plural, but it's not going to change the answer to the question. Does anyone want to take a stab at it? It's okay if you're wrong. This is a confusing question. Yeah. D, yeah. Well, why do you think?
The process of elimination?
Yeah. So D is false because the majority of viruses are smaller than cells, but the key word there that makes this false is always. So this is one of those things that I want you guys to kind of keep an eye out for questions on exams. Anytime in biology when we talk about things, there's always exceptions to the rule. So if a statement says something that's very definitive, like always, that would, should kind of like alert you guys to question that statement. So just wanted to, give you guys that, and it'll be relevant more as we move on. Questions? Okay, so now we have to talk a little bit about the physics of light in order to understand how we're using light to see microbes. And so light microscopy is one of the most powerful tools to study cells. And the reason is because it kind of extends the lens system of our own eyes, and it uses the electromagnetic spectrum. So light is part of visible light is part of the electromagnetic spectrum. And this is all dependent on wavelengths, and the wavelength of visible light is 400 to 750 nanometers. So that's just something that we know. And so microscopy takes advantage of light and how it can interact with matter. So some of the things that I want to just talk about quickly is that light has particle or photon properties and wave properties. We're not going to get into all the details of that for this class, but these properties allow light to interact with matter in different ways. And those different ways are absorption, reflection, refraction, and scattering. So if something is being absorbed, this is the object, the matter. If it's being absorbed, here you can see that in absorption, it's going to absorb something and increase the heat. And so the object here blocks part of the light that's coming in. So the light here is in yellow. Reflection is based on angles and the wavefront of the light, and it's bouncing off of the surface directly. It's being reflected. Refraction is an important one that we'll be talking about more than the others. It's when light hits the surface and then it bends, and so because it bends, it changes its speed, and then when it leaves the surface, it bends back and it changes the speed again. So it's able to go through and it gets refracted. And then there's scattering, where you have the light scattered in all directions when it hits the object. But the one that we'll focus on here is refraction. And that's because magnification on a microscope requires refraction, which is the bending of light rays. So here we're looking at glass, and first the light is coming in, and there's something that we call a refractive index, which basically determines how the light shifts when it hits things with different refractive indices. So here there's a note that says glass refractive index is greater than for air. What you need to know is that the refractive index for glass is different than air. So when the light is in the air, it's coming at a certain angle, and then when it hits the glass, because there's this refractive index, it's going to bend, and then when it leaves, it goes back to that original change in direction due to the refractive index. So whatever I just described here in refraction, this is all due to this refractive index. And so if you have a glass lens that is parabolic, bent a little bit, there's a way that where if light enters it, it's going to bend, but these rays all kind of bend so that they all meet at a certain point called a focal point. And that is how light microscopes work. We have a focal point, and the lens bends, the lens will bend the light rays so that they can intersect at this focal point. So that's how, magnification is dependent on refraction and these refractive indices so that we can see the image on the microscope. Okay, so which of the following phenomena allows a lens to magnify the image? Yeah, refraction, A. So these are sort of also similar types of questions that I'll ask on exams to give you guys an idea of how they look. Okay, so now that we got away from physics, I want to talk about some of the different types of microscopy that we're going to be using in lab. So it's important to pay attention here. One of them is bright field microscopy. Basically, what it does is it generates a dark image of the object over a light background. So here you see that the actual cells are darker than the background. The background, you can see the shine around it to help give it that contrast. So to increase the resolution, you want to use a shorter wavelength, balance the light and the contrast, and then we can use immersion oil and a wider lens. So that would be the lens that would be 40x or 100x. For things that require bright field microscopy, that's the only time that we use the 100x lens in the lab. So basically, the use of immersion oil in bright field microscopy is something that is going to be really relevant next week. So the refractive index is the ratio of the speed of light in a vacuum to the speed of light in another medium. So the refractive index of air is 1, in water it's 1.3, in glass it's 1.5. You don't have to memorize these numbers, but what you should know is that the immersion oil has the same refractive index as the glass lens. So by putting oil onto a slide, you end up compensating for that refraction and the light bending, and so it allows you to see the image more clearly. So using immersion oil will minimize the loss of light by refraction, and it makes the 100x magnification possible with minimal distortion. So here you have a glass slide, and then on this side you have no oil. So you'll see that when the light hits the glass slide, because the microscope stage is down here, and over here is where the light source is coming up. So when the light hits the glass slide, it's going to, with no oil, it's going to refract, and the light is going to be lost. But when you put immersion oil, and the light hits that focal point, the immersion oil has the same refractive index as the glass slide. It's not hitting air anymore, it's hitting the oil. And so this allows the light to then pass through the objective lens. This is the 100x lens, and then you can actually see the image. So here's a little video. So you have oil being poured in a glass beaker. So when the oil is poured in a glass beaker, because they have the same refractive indices, the beaker that's on the inside becomes invisible because you don't lose that light. Does that make sense? Okay. So in lab, you're going to be using these compound microscopes. We also call them light microscopes. And so, we talked about this stuff in lab already. The objective lens, the first magnification step, the ocular lens, which is the eyepiece that's already 10 times magnified. And then we have total magnification. So that's the objective lens multiplied by the 10 times ocular lens. And so one of the things that we're going to do in lab, so that we're also going to be doing things with bright field microscopy using the 100x and the oil immersion, but we're also going to be doing something else. These are going to be wet mount preparations. So I'm going to be giving you a sample of some water from a pond, and there's going to be organisms in there, and so we're going to put those on a slide, cover it with a cover slip, and then we're going to look at it under the microscope to see who's there, what's swimming around. This is called a wet mount preparation. So it's a simple way to observe microbes by placing them in a drop of water on a slide with a cover slip. So here you have a slide, you put the drop, and then you cover it with a cover slip. So you want anything that's wet, that's in a drop of water that you put on a slide, has to be covered with a cover slip so that the water, is contained within the cover slip. There are advantages to wet mount preps, and that's that you can actually look at the cells in their natural state. You don't have to kill them or anything. You can just look at them in that water droplet as they're swimming around. The disadvantage is that some microbes move really quickly, and so it's hard to see them. The sample can dry out quickly, and there's little contrast between the cell and the background. So there's a lot of microbes that are clear, and they're in the environment that's also clear, so without staining it, it's difficult to see. Okay, so when you're trying to find focus under the microscope, you are going to be playing around with the coarse knob and the fine focus knob. And so these are just some examples of what it would look like if you, if you're not focused in too well. So you might see here in A, it's too far beyond the focal point, so it's pretty blurry. You can't really see what it looks like. If it's too close to the lens, it'll look blurry. If it's in focus, it'll be sharp. And over here, if the object is actually not sitting directly like this on the slide and it's actually pointed in a different position, it might skew the way that you're seeing it. So you have to remember that these microbes, are 3D, and so you might have some stacked on top of each other, and so it's hard to actually see the individual cells. So just some things, when you're finding focus to think about. And so the next thing that I want to quickly run through is when we want to look at microbes that are not in a water sample, how do we do that? We have to stain them and you have to kill them. So we call that heat fixation. You're killing them with heat, but you're able to adhere them to the slide in a fixed position. That's why it's called fixation. And so usually we use a heat flame or methanol to do this. And then once you do that, you fix them to the slide, you can then stain them, and then the stain gives them a distinct color. And then we don't have to know the stain and how it works, but these two methods usually will kill the cells. So the first type of stain that we're going to use is a simple stain. A simple stain is just a simple one color, it's dark color that specifically binds to the cells, but it doesn't bind to the external media or the surrounding tissue. Methylene blue is one of the commonly used stains. Crystal violet is another one. We're going to be using crystal violet staining in lab on Tuesday. So just to give you a little bit of just introducing you to what this procedure looks like, I'll have this in lab as well. But what you're going to do is you're going to take a loop full of the culture that I give you. You have these loops in your drawers. You'll put it on the slide and you're going to spread it in a thin film over the slide. And then you're going to let it air dry just to make sure that it's on there, because if you were to just put it through the flame to fix it, which is the next step, then you might just completely, it'll all evaporate and dry off. So you make sure it air dries on its own, and then you do the heat fixing step. So you heat fix it. In this case, they're adding methanol. We'll do a heat fix with the Bunsen burners. I'll show you that in lab. And then after you heat fix, you can stain. So you put a drop of the dye, and then you have to wash off the dye and blot off the excess water. and then you can look at it under the microscope. You don't need a cover slip for this because it's not a wet mount. It's just a fixed stained microbe. So this is the simple staining procedure. Just a reminder for lab on Tuesday, we don't have class on Monday, it's Martin Luther King Day. For lab on Tuesday, you guys have to bring a notebook to class. And I'll have the procedures up before class so you guys can take a look. But yeah, that's all for today.
Oh, I forgot. You have COVID. No, I don't. I tested negative. We have. I wouldn't be here if I was positive. Jesus Christ. Did you blast it?